surrounded by stromal cells, there's blood vessels, there's neural inputs, many soluble factors for the blood and other chemokines, such as GCSF and stem cell factor made by the progenitors and other cells locally. How can we understand that environment to both understand the normal process and disease? So together with a graduate student in the lab, Charles Chan, we developed a new assay to ask, could we isolate the niche itself and be able to grow a niche outside of the bone marrow environment? And the way that we went about doing that was if we took a femur from a 14 or 15-day-old embryo, a mouse embryo. At this stage, this is a very small femur and does not contain any blood cells, but it's the early area where the bone marrow niche is going to form. We could take this tiny little femur, which is about 1 to 2 millimeters in size, you can see it right there, and implant that underneath the kidney capsule of a mouse. The reason we put it underneath the kidney capsule is that's a very vascular environment, and it will support the cells that are in this very small femur. Then we asked the question, what would happen to that femur? Well, if we waited about a month, it went from about 1 to 2 millimeters in length to almost a full-size femur that you find in an adult mouse. It grew over 10 times in length, and you can almost appreciate these pictures here. You can almost see some red color in there, signifying that there might now be bone marrow. If we look in this environment, again, it's just a putative femur. At that stage of development, there's no bone marrow in that environment. We could track these by luciferase positive methods and others and show that these bones would survive underneath the kidney capsule for months at a time. They would even engraft underneath the skin in a subcutaneous manner. Now, what was particularly interesting then is we actually looked at this histologically. It looked pretty much like a normal bone in an adult animal. And if we put those bones, these fetal bones, into a GFP positive animal, so these came from a GFP negative animal into a GFP positive, if you looked inside, they were filled with green cells that were derived from the recipient. The donor environment was from the donor femur, but the cells that were engrafting that came from the recipient or the host. And the question now, what were these cells? Well, it turns out these were actually hematopoietic stem cells. So we took a normal bone marrow from a normal mouse, and we look in the bone marrow using flow cytometry. We can identify the hematopoietic blood stem cell. It's c-KIT positive and SCA1 positive. It's lineage negative, has some other characteristics. And we can find that population. If you look at the kidney, normal kidney, you won't find it. But if we looked in that engrafted bone, we'd also find that phenotypic population of hematopoietic stem cells. We could then transplant those, take those from that femur right in here, take the cells out and transplant that now into a mouse that was irradiated, and look for donor-derived GFP positive cells that we'd find over weeks to months, the generation of donor-derived cells, like indicating again that this engrafted femur was a new bone marrow environment and actually contained hematopoietic stem cells. So that was all well and good, but we're not going to be transplanting full femurs anytime soon, certainly to people. We want to understand, could we actually reconstitute the niche from the cells in that femur itself? What were the cell components that could create the niche? What are the niche stem cells, essentially? And to answer that question, we started basically by first just digesting these femurs using collagenase and a few other methods. We could then take that whole mix of cells and put that in a little matrix and implant that, again, underneath the kidney capsule. Alternatively, we could also, as I'll describe later, sort specific cells from that heterogeneous mix. So if we took, let's say, a simple femur from a 14-day-old embryo, digested those cells and implanted them underneath the kidney capsule, when we looked about a month later, we would actually find generation of a new bone marrow environment. Again, in this case, it wasn't a femur, but it was actually a complete bone marrow environment. If you touch this, it's hard, it's crusty, it feels very different than the kidney itself. You can see there it has some volume to it. These are different examples. And if we looked in that on the surface, it looks, again, like bone. It's vascularized. And if we look at it histologically, it looks essentially identical to bone marrow. This is from the intact femur that we transplanted earlier, and you can see what looks like trilineage hematopoiesis inside the bone marrow. This is the one from the dissociated cells, only from 1,000 to 10,000 cells, would recreate this entire bone marrow niche, including the cartilage and bone formation around with the bone marrow environment in between. And these cells, again, come from the circulating hematopoietic stem cells of the recipient mouse. So we can actually look at that on CT scan. We can find this little calcified cap here. You can see it again on CT. So this is, again, a new bone marrow environment created underneath the kidney capsule. and seems to function like bone marrow. If we wanted to look at the time course, how long would this take? Basically, the bottom line, it would take about 32 days. If we look by day 32, we look for hematopoietic stem cells in that cell environment, we start to see them at around day 32. Whereas if you looked at the control kidney or normal and compared to normal bone marrow, it's essentially identical. So it takes about 3 to 4 weeks to generate that new bone marrow environment. And those cells would engraft into a recipient at about the same or better than normal bone marrow. Now the next question was, could we identify the true stem cell niche progenitor? So we would take that femur, digest it, and sort different populations. I don't have time to go into all the details of the flow cytometry, but we picked several populations that we thought would be high yield, and we looked to sort for cells that were CD105 positive, which is a marker for mesenchymal stem cells. We'd look for alpha-5 integrin positive, and we'd sort for either 105 positive, negative, and then Phi 1.1 positive or negative cells. So we had basically 3 different populations, and we could sort those cells and transplant each of those separate populations underneath the kidney capsule. And the bottom line was only one population, the CD105 positive, Phi 1 negative population would form a complete bone marrow environment, as you see here, whereas the 105 positive, Phi 1.1 positive population would only form bone with no marrow. And if we use 105 negative, Phi 1 negative, we'd basically get nothing. So we could differentiate between this heterogeneous population, a specific population of cells that was quite rare, that would recreate the hematopoietic stem cell niche. If we looked in these histologically, we could see that the blood supply came from the host, again, as well as the hematopoietic stem cells. And if we transplanted the purified stem cells that came from those grafts as well, those would rescue a lethally irradiated mouse. Again, that's the standard approach to prove that you can rescue and reconstitute that animal with the donor-derived cells. So, in summary for this part, we were able to identify a pure population of cells from the fetal bone, and that population, by the way, does exist in adult bone at a lower frequency. It could recreate the niche as well as support hematopoiesis. The next question is, could we start to manipulate that niche and understand what factors were important in supporting and generating that niche? And there are several factors that are known to potentially play a role in both hematopoiesis and bone formation, including steal factor, Asterix, and VEGF. And we can knock those out in various ways. We could use silencing shRNA and look, for example, in the setting of to knock out Asterix. In this case, there was no impact in that setting, or probably for steal factor. That factor did not seem to play a role. We still get the same bone marrow formation. But if we used Asterix, which is involved in bone formation, in general, that would inhibit the niche formation. So again, the mock infected inhibitory RNA would enable a full marrow graft, whereas if you inhibited that, it would knock out that process. And not surprisingly, VEGF, which is very involved in blood vessel vascularization, if you knock that out, you would just end up with a little bit of cartilage, but with no bone marrow environment. So an example of trying to manipulate the niche and understand the factors involved in its generation. So in conclusion for this part, we have a new assay for creating a bone marrow environment outside of the bone marrow arena. We've identified a population which in detail is CD45 negative, Tie2 negative, alpha-5 integrin positive, 105 positive, Phi 1.1 negative. We can sort those cells purely, and those cells in very small numbers can create a new stem cell environment. And we've been able to start to identify the factors that play an important role in that process. So how might this apply to aging? Well, we know that the bone marrow environment changes over time from a young donor, if you do a bone marrow biopsy, to older patients which have a much less cellular marrow. It's filled with more fat. It generally has less hematopoietic stem cells. And there's been a lot of work now. If we look here at the formation of that area, again, with VEGF and others involved, that niche may be quite different in older patients or older individuals. So the question is, by manipulating the niche itself, can we essentially improve the environment for those blood-forming stem cells and potentially have a model for other stem cell type conditions? We do know, and work done at Stanford by Derek Rossi and others, that older hematopoietic stem cells, when we transplant those into recipient mice, do not act the same as young stem cells. They make far more if we take